All right. Next up, we have Sandy Macrae, President and Chief Executive Officer of Sangamo. So a lot going on at Sangamo, Sandy.
Thank you. There's not a lot going on if I don't get the clicker. So thank you. I'm delighted to be here. My name's Sandy. I'm the CEO of Sangamo, where I've been since June. And of course, we're going to talk about some forward-looking statements. And as many of you, hopefully all of you know by now, we're now Sangamo Therapeutics. We decided to change our name. We decided we had to celebrate Sangamo because Sangamo has been around for over 20 years and it is the scientific heritage of the company. But we decided to move from bioscience and a platform-based name to therapeutics, reflecting the fact that we're moving into the clinic and underlining to my staff and to you the importance we feel about treating patients and the responsibility we feel to move our science forward and translate it into groundbreaking medicines that will benefit patients. And underpinning that has been quite a fundamental change at the company, with the final piece of that yesterday when we announced that Edward, who was our founder CEO and then became our chairman, will not be standing for re-election. So there's a complete change within the Sangamo leadership. But underneath that are putting together the pieces that allow us to move the company forward as a therapeutic company. New CFO, new CMO, new head of manufacturing, hiring the right clinical people, putting in the right processes that allow you to move reliably forward to the clinic. And our priorities for the coming year are very, very simple. Number one is the only one that you need to care about. Number one is our priority. It is to make sure that our 4 clinical studies advance into patients and we can demonstrate the benefit of our technology. But behind that, we're going to continue to advance the platform. I'd be delighted to answer questions on it because I do think our platform is not fully understood and truly is best in class in gene editing. And because of that, we think that the move now for companies like ours is not to be better and better at gene editing, but is to be part of the ecosystem of editing delivery, because it's where you can put it and how you can put it easy into humans. And so whether it's LNP or new forms of vector, we're being part now of the scientific advances in that, as well as with editing. And finally, of course, partnerships are important. We're a company of about 140 people. We can't do this alone. And so we're going to be much more open than my predecessor's company was to partnering with companies that understand our science and share our values about bringing medicines to patients. So we have 4 clinical trials. We have 2 of them that are now open and recruiting. We have, if I go from top to bottom, hemophilia A is gene therapy. It should open in the next, this quarter, the second quarter. We have the IND open, we're ready to go. Hemophilia B is up and recruiting. The sites are open and we are looking for patients. MPS 1 and MPS 2. MPS 2 is open and recruiting, and MPS 1 will be open, as it says, imminently. So we're fulfilling what we said we would do, which was to move forward with simple clinical operations and clinical studies, and we're delighted to see that these are all working. And remember, it's not just about the first patient, it's about the second and the third. It's not just about the first site, it's having a pipeline of sites so you can ensure success and ensure proper recruitment. So this is the slide I'll stop on. This is our pipeline. And what I've tried to do in the short time I've been at Sangamo is focus down our pipeline and balance it so as we have the 4 things that are now in clinical development. And behind them, we have our partnership with Bioverativ for beta thalassemia and for sickle disease. We're very proud of that partnership and the work that the team in Bioverativ are doing with us. And we're just completing the IND-enabling studies, and very soon we'll be moving forward with that IND. We have a wonderful technology for oncology, for T cells, I think is best in class, with editing rates that no one else is achieving, and we're looking to partner that one out. We have Huntington's disease with Shire, and we have delivery. But the one that caught a lot of interest in the past month was tau. We were very fortunate that Biogen chose to buy Alzheimer's programs from BMS because it came at the same time as the announcement of our ability with our zinc finger transcription factors to edit tau. And we can control the production of tau and almost shut it completely off. And that, I think, will be one of the lead programs that moves forward in partnership with someone else as a new generation of zinc finger capabilities. So I'll stop there and gladly take your questions.
Thanks for the overview. Having watched Sangamo for many years before you joined, it seemed like a lot of the progress was actually circular, as opposed to forward and linear. And maybe looking back at the history of the company as you're joining and now pushing forward, what do you think that's attributed to? Did the technology need to evolve? Did the delivery routes need to evolve? Did we need better understanding of targets or better strategy for target selection?
I see we're starting with the easy question.
You bet.
So let me try and be philosophical about it, because it isn't, I haven't come and waved a magic wand at Sangamo. I think there's something that's unappreciated about the adjacent technologies had to evolve to make gene therapy and gene editing easier. And it's not just about delivery, it's about how easy it is to manufacture, how easy it is to sequence, which we do to make sure we can reduce off targets. So all those things have come along and made life easier for us and made it easier to move into clinic. I think our friends in the CRISPR company have created an energy in the field and an investment in the field that has reminded us all that we're going to move forward in gene editing. I'm responsible for scientists at Sangamo, some of which have been there for 20 years. They've given their whole life to this technology, and it really has been a lifetime's dedication and work to perfect the technology, which is why it now is best in class. They didn't always make the wisest decisions of which indications to go for, and that's because they were a very scientific company and didn't always have the right clinical people to give them advice on what clinical indications were best to go for, and commercial people to talk about patient need and the market ahead. And so one of the first things we did is we looked at where the medical need is, and I think if you're going in with a technology that is cutting edge, what you want to do is choose indications that are proven and that you're reducing the number of variables in the experiment. And to me, hemophilia and the mucopolysaccharidosis are perfect examples of that. A previous study that was done at Sangamo was in diabetic neuropathy, and any of you that have done a diabetic neuropathy study know that it's a difficult indication, it's difficult studies to do, the patient population is very diverse. So even if the product had worked in that, it would have been difficult to prove it. Whereas it's very easy to know that you've been successful in hemophilia, and you will be able to measure success in the mucopolysaccharidosis, and the medical need there is enormous, because the enzyme replacement therapy is beneficial but not curative. And if we can move first into adults and prove that we can, by dropping a new gene into the liver of these patients, and this is the bit that I get this frisson every time I talk about this. The Sangamo project makes a cut with an intron 2 of the albumin gene and drops in a new transgene. So it's genome editing rather than gene editing. And the albumin promoter drives the production of enzyme from this locus and replaces the enzyme for the patient, for the child, from a site, a safe site within the liver. If we can prove that that works in adults, our aim is to take that back into children. So as eventually, I have a dream, I really have a dream about this, that we will be able to diagnose children with these enzyme deficiencies very early on, do a simple 30-minute infusion or 60-minute infusion, and then they will be, their condition will be addressed for life. So it isn't a cure, but it's the eternal replacement.
And you mentioned some of the relative advantages of zinc fingers. Can you elaborate on that?
Yeah, and I was at a meeting yesterday that was sponsored to look at standardization of gene therapy and gene editing. And one of the things it revealed is that there isn't a common lexicon for how we describe editing. And one of the things that the group's going to do is start to codify and clarify how you think, what you think about when you think about editing. And so the way I think about it is, the first one is efficacy. Are you able to cut wherever you want to do within the genome? And zinc fingers don't have a guide sequence, so we can cut wherever we want. And with even what we've got within the cupboard in Point Richmond, we can cut at 60% of sites within 10 days of looking for designing a set of zinc fingers. And with a couple of months of making bespoke zinc fingers, we can get that up to 100%. So there is nowhere within the genome that we can't cut. We aren't limited by the guide sequence. That's one. The second is how effectively you cut. And in all of the tests we're doing, we're now up to 80 or 90% efficiency. So you create indels at 80 or 90%. The third is what's the off-target. And how you measure off-target is important. You can measure it in a biased way, which is really bioinformatically predicting where you might cut off target. Or you can do as we do, where you actually do the experiment and you sequence, using the new sequencing technology, across all the places you actually did cut. And using that more advanced technology, we can show that we can get it down below the levels of detection, that the off-target is now at the levels that we all get, because we all have cuts within our genome. When you, particularly as much as the audience here flies, there's constantly double-stranded breaks happening in your genome, and we can get down to levels that are indistinguishable with that. And that was some wonderful work done by a man called Ed Rebar, who is the lead scientist on zinc fingers and has been with the company for 18 years, I believe now. And his discovery that it's not just the fingers that are interacting with the DNA, but it's the backbone, the beta-pleated sheet, that was having non-specific binding, and that by adjusting the charge of that, he could remove the off-target. And I tell a little bit more about that story because it's pleasing in that it's logical and it's something we now have mastery over rather than just a result that we've got. So now going ahead with this cupboard full of zinc fingers that we have, where we can produce zinc fingers within 10 days in 60% of the sites, we can now also keep the off-target low in a predictable way. And then the final things I would think about when I think about editing, one is, can you do it in the tissue of choice? So when people report this, be very careful to see that they're not doing it in an Eppendorf, but they're doing it in the tissue that they'll eventually go to edit, and that they're doing it at the right scale. Because it's easy to do it in small scale and harder to do it at clinical scale when you've finally got a 400-litre or 2,000-litre production run. And that's how we're reporting our editing. And the final one is, can you deliver it to the tissue of choice? And that goes back to what I was saying earlier, that once you've mastered editing, it's all about delivery. And it's been very interesting to hear some of the presentations today about delivery. And I think that will be an ecosystem of partnerships between the different companies here.
Any questions in the audience? And if not, what about the delivery that you're choosing? Going with an LNP may allow you to repeat dosing. Is that part of the strategy, or is the view that the zinc finger cutting efficiency and delivery through LNP can suffice for one shot?
So our current programs are all with AAV6, and I was interested in the previous speaker's comments about neutralizing antibodies, and perhaps would disagree a little. We've taken the time to look at the neutralizing antibodies for all of the vectors, because I think doing it all in one system is really important. We're also being very careful to look not just at the concentration of neutralizing antibodies, but their impact on the transfection. And we're very happy that the majority of patients, 60% of patients would be available to, or more would be available to AV6, and that we're looking for the lowest titer ones to ensure the cleanest clinical results. But ideally, one would like to go beyond that to LNPs for several reasons. One, you avoid neutralizing antibodies. And the second, you can repeat dose. And as a clinician, thinking about how I would dose a patient, that allows a far more exquisite control of how much you give of any of our products. Because in the end, that's what we want to do. We want to titrate it to effect and give the patient the best dose.
I like what I'm hearing from Sangamo. Thanks very much, Sandy.